Thank you for coming. This is our work. That work was performed by reproduction group and supported by Universidad de Antioquia and Universidad Cooperativa de Colombia. The name is the effect of sialic acid expressed by syncytotroph blood cells on immunomodulation of peripheral blood cells. Sialic acids are terminal monosaccharides of glycan chains and glycoconjugates and play an important role in cell recognition and the regulation of multiple physiological processes through binding lectin-like receptors. During preeclampsia, we demonstrated that NK cells were modulated and the production of both anti-inflammatory and pro-inflammatory cytokines are higher compared with the controls. Other cytokines are higher, for instance, vascular endothelial growth factor, and we propose that this immunomodulation is induced in the maternal-fetal interface and possibly by the expression of some glycans in the syncytotroph layer. The objectives of our work were to evaluate the expression of sialic acid in syncytized vivo cells and to evaluate the interferon gamma expression by peripheral blood cells co-culture with vivo cells and the relationship with sialic acid recognition. We stimulate vivo cells with phoscolin and syncytize these cells and later co-culture with peripheral blood mononuclear cells. And finally, we tested the expression in both subsets, thin and bright NK cells of interferon gamma. And in other set of experiments, we stimulated the syncytized vivo cells with TNF and interleukin-1 beta, or with a serum for preeclamptic women. We found that the production of interferon gamma by bright NK cells were lower when these cells were co-cultured with vivo cells, and we restored the production of interferon gamma when we remove the sialic acid in the syncytized vivo cells, and we compare the expression of this sialic acid and remove the sialic acid with a cellulose treatment, and we observe the differences between the production of interferon gamma. If you can see, the interferon gamma production was higher in the NK bright NK cells subset compared with DIM. And in the other set of experiments, we demonstrated that pro-inflammatory cytokines, mainly TNF, induce a higher expression of sialic acid, and this effect is almost the same in the set of experiments in which we stimulated the sialic acid expression with a preeclamptic syrup from with a syrup from preeclamptic women. The conclusions of our work were that NK cells co-cultured with vivo cells, namely bright subset, were negatively modulated for interferon gamma expression. Sialic treatment increased the NK bright positive interferon gamma positive cells. Syncytized vivo cells expressed higher levels of sialic acid when they were stimulated with recombinant TNF or with a serum from precancerous. Thank you for to be here. Bye bye.